Okay, cool. Assalamualaikum. All right. Okay, make sure you have what we call laboratory guidelines. Okay. MIC 180. Okay. So now we are doing fully 100% ODL. So which means that on the week number 3, actually, we can do the laboratory qualitative test for amino acids. All right. From here, okay. All right. For laboratory executions, this is to be applied for all lab topics except for lab number 4. Why lab number 4? Lab number 4, you have to do your own lab reports. Okay, the rest you'll be just briefing for the labs. Okay, so they are what we call short quiz for test student understanding. Okay, and complete the quiz and consider the attendance for the lab session that week. Okay, ni maksudnya dekat sini adalah awak kena siapkan post soalan yang saya akan bagi dalam Google Form tu. Tolong hantar within pukul 4 ni. Approve for your attendance. Dan juga discussion dalam in future. Okay, please note for lab number 4, you have to do your own lab report individually. Okay, so maknanya lab 4 saja yang akan dimasukkan dalam markah. Okay, sebanyak 20 markah. Okay, so the rest will be on attendance. Okay, so there what we call video reference for you all to see. Okay, for the lab 2 qualitative test for amino acids. So please do video 1 and video 2 ni. Okay, so check video apa yang ada ni. Alright. Alright, so we go for the today's experiment. Okay, so eksperimen hari ni kita akan buat eksperimen number 2, qualitative test for amino acids. Alright. Alright, so laboratory number 2, qualitative test for amino acids. Introduction. Amino acids are the building blocks of proteins, which means it's a monomer for proteins. So protein is our polymer, so monomer for the protein is our amino acids. They share the same acid and amino groups, but the site change differ. Okay, when the amino acids are linked together to form an amide, the product is called a peptide. And the newly formed bond is called a peptide bond, right? If a few amino acids, we call it up to 20 are linked, we call it polypeptide. If we say more than 20, it's about 20 to 100 link of amino acid, we call it as a proteins. Okay, so that's meaning of protein. Okay, all right, we go for the experiment number 1. It's an ninhydrin. We're using an ninhydrin. The alpha amino acid can be detected by using ninhydrin, which form a blue violets. Okay, color as a positive indications. Other than that, selain daripada warna blue violet tu yang awak dapat, let's say yellow, orange or red, it consider negative. So why we need to do this ninhydrin test? Untuk mendapatkan bahawa alpha amino acid. Untuk detect alpha amino acid, alright. So mestilah berwarna blue violets. Okay. The test is very sensitive, often used for colorimetric determination of amino acid solution or as a visualization agent in the chromatography of amino acids. Okay. During the reaction, the amino group will liberate ammonia. Okay, so what happened to ammonia? Ammonia will react with the ninhydrin and form a colored complex. So colored complex can be yellow, orange or red, even negative. If it's positive, blue violet. So actual color for ninhydrin ataupun warna asal dia adalah warna biru. Okay. So number 2, the most protein give positive result in Xento protein test. Why? Because the presence of phenyl groups, C6H5. Okay, so this phenyl group produce colored nitro compound in the reaction with the nitric acids. Okay, so eksperimen kedua adalah test yang kedua adalah kita gunakan Xento protein test. Okay, number 3, the amino acid with the thiol groups or SH, sulfur S represent the sulfur, such as cysteine is one example of amino acids. When heated in the presence of alkali, will produce sulfite ion. These ions can be detected by reaction with the lead cations. A black precipitate plumber sulfite is formed. Okay, bila campurkan dua-dua bahan ni, kita campurkan salah satu amino acid ke dalam ni dengan sulfite ion, dengan apa tu bahan seperti plumbum, kita akan dapat black precipitate. Okay, sebab apa dapat black precipitate? Because all amino acid have a thiol groups. Contohnya cysteine, right? So juga amino acid yang lain. In Milan test, if the hydrophenyl side group is present, a red color will be observed. Tyrosine is the only amino acid which give the positive test. So kita akan pakai amino acid nama tyrosine saja. So tyrosine ni akan berikan jawapan yang positif pada Milan test tu. However, any molecule with the phenolic OH will react juga. Tapi the best positive result kita gunakan tyrosine untuk Milan test. Okay. In Hopkins-Cole test, heterocycle side change or indole change of tryptophan react to give a purple to violet rings at the interface of the tower to layers. So masa saya start buka kita punya Google Meet tadi, awak akan nampak contoh-contoh result yang dapat daripada eksperimen ni. Salah satunya adalah kita akan dapat purple violet rings. So bila dapat purple violet rings tu, itu adalah Hopkins-Cole test. Kenapa? Because presence of heterocycle cyclic side change. Okay, so amino acid yang kita gunakan mostly untuk positive result, kita gunakan tryptophan. Okay, last one will be the Sakaguchi test. So mostly Sakaguchi is ammonium ion give the positive reaction in this test. So mostly kita akan dapat warna pink ataupun merah. Okay. Okay, we go for the objective to conduct a series of qualitative tests for amino acids. Okay, alright, method dia, test 1, anhydride test. Okay, we put 1 ml of glycine in the test tube, 5 drops of anhydrines, mix it up, and then boil on the boiling water on the bottle bath for 2 minutes and record the changes. And then repeat with the others amino acids. Okay, number 2. Xento Protic test, we put 1 ml of glycine or tyrosine, tryptophan, phenylalanine, and phenol. So ini ada contoh-contoh amino acid kita guna. And then put 1 ml of concentrated nitric acid. Swill the test tube and immerse them in the cold water to cool them down. So, and then record the first changes dulu. After that, put 40% sodium hydroxide. And then change the pH solution to alkaline. Okay, maknanya kita akan pakai pH meter and record the changes. Okay. Number 3, Milan test. You have to prepare 1 ml of glycine in a test tube and 5 drops of Milan reagents. Heat the test mixture and boil water bath for 10 minutes. Cool it down and then put about 5 drops of natrium nitrates. And then record the changes. Okay, so use others amino acid as well, yaitu tyrosine or phenylalanine. Hopkins-Cole test, you have to mix 1 ml of glycine, one example of amino acid, with the 1 ml of glycol acetate acid in a clean test tube. You have to incline. So macam kita nak buat incline, incline awak kena gerakkan 45 darjah test tube awak tu dan masukkan secara perlahan kedua-dua mixture ni. Mula masukkan glycine dulu, lepas tu masukkan 1 ml glycol acetate pula ke dalam test tube tu. Make sure jangan bercampur. And then last one, add 1 ml of concentrated sulfuric, but do not mix. Okay, so awak akan dapat 2 layer should be formed. Okay, and then let the layer stand and note the color at the interface between 2 or 3 minutes. Repeat using stock solution in other of amino acids such as tyrosine or tryptophan. So dalam eksperimen Hopkins test, positifnya adalah tryptophan. Okay. Alright, number 5, lead sulfite test. Pipette 1 ml of cysteine in the test tube. Add 1 ml of 40% sodium hydroxide. Swill the test tube. Heat the test mixture in the boiling water about 2 minutes. Immerse it with the cool water. And then put it 1 ml of sodium palambate. Okay, and record any changes or the presence of black precipitate. Okay, repeat using stock, other stock ataupun repeat other amino acids such as cysteine and methionine. Last one will be your Sakaguchi test. Okay, put 1 ml arginine in the test tube. Put 1 ml of 40% of sodium hydroxide, 2 drops of naphthol, 5 drops of bromine, and then swill the test tube. Okay, and then record your changes. So repeat the procedure by using other substances such as glucosamine, methyl guanidine, creatine, and last one will be urea. Okay. Right. So if you do the experiment, you have to put it on the table. For example, you had retest, we have a lot of sample here, glycine, tyrosine, tryptophan, and phenolene, and protic. So this example of is what we call qualitative test. Why we call it qualitative? What is the difference between qualitative and quantitative? Qualitative more on the process of observation. Example, the unit using is the changes of the colors. For example, kalau glycine, so tadi adalah from blue to blue violet. For example, you get the glycine, result dia blue to blue violet. So the conclusion will be presence of alpha amino acid. So blue to yellow, which means not present of alpha amino acid. So that's meaning of qualitative and quantitative. If it say about quantitative, it's more on the measurement. For example, length, in kilometer or meter. Okay, something that you have to do the measurements. Okay, that's the difference between qualitative and quantitative in your observation on the practical lab. Okay, in science. Okay, you can put all the result inside the tables and then you do the conclusion, right? Okay. Right, is there any question before we start another PowerPoint? Ada soalan tak? Is there any question?
Sir.
Yeah. Yes.
Yes, I want to ask. Means our observation,
Okay.
changes of color, macam tu ke?
Observation of what? Maksud awak?
Kalau macam changes of color of the substance.
Okay, substance that one is more on qualitative lah. Kalau changes of color refer to our qualitative. Kalau apa tu kuantitatif, you all kena measure lah. Dia kena ada unit. Macam ada nombor puluhan. Itu adalah kuantitatif. Difference between quantitative and qualitative.
Okay. Thank you, sir.
Alright.
Alright.
Okay. Is there any question there? Tak ada. Alright, so continue. So this one is more on chemical reaction inside the qualitative. Since kita tak boleh nak buat lab ni, so saya akan ceritakan kenapa berlaku perubahan warna tersebut dan contoh-contoh amino acid yang akan berlaku dekat mana structure dalam kita punya chemical tu. Alright. Okay, let's say what is amino acids? Amino acids are the building blocks of all proteins. Are linked series by peptide bonds. Okay, one amino acid bond with another amino acid dipanggil sebagai peptide bond. Ini betul CON and H to form the primary structure of proteins. Okay, passes an amine group, a carboxyl group, acid group, and a various side change that differ between different amino acids. There are 20 natural occurring amino acids which vary from one another with respect to their side chain. Okay, this is our example one of the amino acids. There are two of functional group here. Number one will be amine group. Okay, kita tunjuk ni. So amine group akan memberikan basic. Okay, another one will be carboxylic group. So this one will be your acidic part. Okay, so yang ayat terakhir ni, kenapa ada 20 jenis? Because of presence of side chain dia, ni R ni. That's why kita ada lebih kurang 20 amino acid. Sebab presence of R group ni. Right? So bila ada kedudukan macam ni, barulah awak tahu bahawa itu adalah amino acids. Right? Okay, for today's experiment, okay, saya akan ceritakan tentang chemical reaction ya. Kenapa berlaku perubahan warna tersebut. From ninhydrin until the test for presence of cysteine. So result color tu awak boleh check dalam video. Alright, we go for ninhydrin test first. Ninhydrin degrades amino acid into aldehyde, which is pH range number 4, ammonia and carbon ester through the series of reactions. Okay, and then the reduction products obtained from ninhydrin, what is called hydridantine, then react with the ammonia. Okay, ammonia ion, excess ninhydrin to produce an intensive blue or purple pigment. Okay, so bila dapat blue atau blue or purple pigment tu, maknanya ninhydrin test adalah positif. The amino acid proline and hydroxyproline also react with the ninhydrin, but they are giving the yellow color complex instead of purple one. So kita ada satu jenis amino acid amino acid tu nanti masa kuliah awak boleh check, dia akan react juga dengan kita punya ninhydrin, tapi dia hasilkan bukan warna biru ataupun purple, dia lebih kepada warna kuning. Okay, alright, we have a ninhydrin and then we have a one amino acids. Okay, bila ninhydrin bergabung dengan amino acid, kita akan dapat hydridantine. Ni betul dia, right? So this one akan bergabung lagi ninhydrin dalam yang cair tu, dan juga dia akan bergabung dengan satu lagi kumpulan amonia. Okay, amonia. So kita akan dapat Riemann purple. So this Riemann purple ni lah yang memberikan warna positif pada kita punya ninhydrin test. Right? Xento Protic acid test. The test gives the positive result in amino acid carrying aromatic groups, especially in the presence of tyrosine. So tyrosine is one of the amino acids dan dia juga ada aromatic groups. So untuk kita tengok positive result, kita gunakan tyrosine dalam eksperimen Xento Protic acid test. Okay. So others than tyrosine, kita ada juga phenylalanine, tryptophan juga akan memberikan result yang positif. So apa yang berlaku kat situ in the presence of concentrated nitric acid, the aromatic phenyl ring is nitrated to give a yellow color, nitro derivative. So maknanya kalau kita buat eksperimen ni, kita dapat warna yellow, result kita adalah positif dan betul. Okay. At the alkaline pH, the color changes to orange due to ionization of the phenolic group. Okay. So let's say kita ada satu amino acid, the example of tyrosine plus HNO3. HNO3 apa dia? Adalah nitric acid. So bila kita gabungkan dua ni, kita akan dapat satu bahan dikenali sebagai nitro derivative dan kita akan dapat yellow color. So all positive result for Xento Protic acid test, kita akan dapat warna yellow, right? Yellow in color ataupun kuning. Bila test, the test will test the presence of hydroxyphenyl side group. Okay, so once you do the Milan test, you get the red color. Okay, maknanya dia adalah positif. So mostly kita pakai tyrosine untuk bagi result yang positif. Or any molecule with a phenyl OH, phenolic OH will react with the experiment ni. So warna dia lebih kurang warna merah. Okay, let's say kita ada tyrosine gabung dengan HNO3, kita akan dapat nitrated tyrosine. You put a Milan solution, kita akan dapat warna lebih kurang warna merah. So Hawking cold test. Hawking cold is a reagent only react with a protein containing tryptophan. Okay. So once the tryptophan is free, it reacts with the glacial acetic acid to form the violet product. The protein solution is hydrolyzed by the concentrated sulfuric acid at the solution interface. Now kita akan dapat warna violet, purple, right? Purple ring. So let's say kita ada tryptophan, kita campurkan dengan formaldehyde. So kita akan dapat colorful condensation product dalam bentuk purple violet, right? Ataupun violet rings. Sebab rings sebab kita letak dalam test tube. Sakaguchi, under the alkaline condition, alpha naphtol. Apa itu alpha naphtol? It's the 1-hydroxy naphthalene. This one will react with the mono substitute guanidine compound like arginine. So arginine is one of the amino acid, which upon treatment with the hypobromide or hypochlorite produce a characteristic red color. Okay, so kita arginine, kita masukkan NaOH ataupun sodium hydroxide plus alpha naphtol. Okay, ataupun 1-hydroxy naphthalene. We mix and then you will get a red color complex. Okay, right? So make sure in the alkaline condition. Okay, last one will be test for presence of cysteine. Okay, used for the detection of amino acid containing sulfide hydryl groups. Okay, ataupun SH group. For example, kita using a cysteine, right? So positive result is a black precipitate. Okay, how? This is cysteine. This cysteine akan react with the, akan dipecahkan menjadi sodium ataupun S2. So this S2 akan bergabung dengan plumbate tadi. So mendapatkan warna black precipitate. Okay, sodium plumbate, right? Okay, so for the process of qualitative ataupun you can see the video to get overview of the qualitative. Video yang saya bagi dalam yang dalam guideline tak boleh guna. Alright, even though awak boleh klik pada awak punya YouTube. Okay, just click qualitative test for amino acid. There's a lot of video there. Right. Okay, is there any questions? Ada soalan tak? Okay, ada soalan? Is there a question for you about this today's experiment? Okay, ada soalan lagi tak?
Tak ada, sir.
Ada? Alright.
Tak ada.
Okay. So, harap semua faham.